by the National Science Foundation, America's investment in the future. By the American Society for Microbiology, and by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
It's no secret that genetic information is passed on from generation to generation. I mean, take a look around. The passing on of DNA in plants and animals is achieved by a mixing of genetic information, which occurs as a result of reproduction. And it's easy to see the genetic traits that organisms share. Microbes pass on genetic information too. While their sex lives are quite different from ours, their DNA is nonetheless successfully regenerated. Human beings, along with other complex life forms, transmit their genes only to subsequent generations. So-called vertical gene transmission means that genetic information is passed on only through the reproductive process. Microbes do this too, but in addition, they have another extraordinary ability that is a far more powerful transmitter of genetic traits. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore the three main mechanisms for microbial genetic recombination. That's transformation, transduction, and conjugation. And we'll also learn how humans have harnessed these mechanisms and applied them for the benefit of all of us. Unlike you and I, or any other creature you can see here at the fair, microbes can pass their genes not only to their offspring, but also laterally to other microbes of the same generation. This is known as horizontal gene transmission, and it's a trait that really sets the microbes apart from multi-celled life. To learn more about horizontal gene transmission, I'm here at Oregon Health Sciences University to meet with Dr. Maggie So, chair of the university's Molecular Microbiology and Immunology Department.
I'm interested in how these microbes can acquire virulence traits. And many of these characteristics can be acquired, can be rapidly spread among a population of bacteria through these mechanisms. The microbes in your body might be totally harmless until they acquire these traits through horizontal transmission. And by acquiring these traits, they can suddenly become more virulent. They can acquire drug resistance, for instance. So if they cause disease later, they would be harder to treat.
So is there a disease that's a good example of this horizontal transfer process?
Well, a good example of this is diphtheria, which is a disease we don't see anymore because of childhood immunization programs. The disease is caused by a bacterium called Corynebacterium. And for the bacterium to cause disease, it has to produce a toxin, which kills the cells in your body. And the gene for the toxin is transmitted to the bacterium via a bacteriophage. And a lot of toxin genes are actually transferred to bacteria through the process of phage conversion.
Phage conversion can result from transduction. Genetic material passes from one bacterium to another via a bacterial virus called a bacteriophage. the virus injects its DNA into the bacterium. The bacterium then becomes a virus factory, replicating the viral genome, making viral coat proteins, and eventually releasing hundreds of new bacteriophage.
Now, every once in a while, during replication of the viral genome, a small piece of bacterial DNA gets inserted into the coat protein as well. And the live virus now transfers this genetic information from the recipient bacteria to a new bacterium.
In conjugation, another form of genetic transfer, a donor cell comes in contact with a recipient cell by way of a conjugation tube, known as the sex pilus. When contact is made, a single strand of DNA containing the genetic information goes through the pilus from the donor cell to the recipient cell. This single strand of DNA is usually in the form of a plasmid, which is an extra chromosomal, independently replicating piece of DNA. A second strand of the genetic information quickly gets synthesized in both the donor and the recipient cell.
So ultimately, both cells now have complete copies of the genetic information that was transferred.
So what's the significance of that from the perspective of someone studying disease, such as yourself?
Well, we now know that a lot of genes coding for drug resistance, antibiotic resistance, reside on plasmids. So a single bacterium in a population harboring a conjugative plasmid containing an antibiotic resistance gene can ultimately spread this genetic trait through the entire population of bacteria, and this could happen quite quickly in the lab.
In transformation, a wide variety of bacteria have the ability to bind free-floating DNA from their immediate surroundings. This DNA may then be inserted into the chromosome and become a piece of permanent genetic information. Bacteria that acquire DNA in this manner inherit the genetic traits that are encoded in the incoming DNA.
How is it that there's free-floating DNA in their environment?
Well, many bacteria have, in addition to their growth cycle, a lytic cycle. That is, they die, their cells burst, and their DNA is released into the surroundings. And living bacteria that are in the neighborhood, if they're competent for DNA transformation, will then take up this DNA. Now, there are bacteria that are very indiscriminate in the kind of DNA that they take up, such as streptococci. And then there are other bacteria that are more particular, such as Neisseria and Haemophilus. And they'll only take up DNA from their own species.
There is another highly specialized form of DNA transfer known as transposition. Pieces of DNA called transposons hop from one genome to another, and sometimes from one region of a genome to another region of the same genome. These self-sufficient transposable elements may code for antibiotic resistance.
So let's say we have a transposable element that codes for a drug resistance gene. This transposable element hops, say, from the chromosome to a conjugative plasmid, all within the same bacterium. This bacterium now makes contact with a female cell, and the conjugative plasmid gets passed to the female cell. Well, the transposable element now ultimately gets passed to a second bacterium via conjugation.
Okay, so it's sort of a two-step process.
That's right. A transposable element can't transfer itself from bacterium to bacterium. It always needs an intermediary.
Knowledge of how bacteria transfer their genes can be used for our own benefit. For instance, not only can we transfer genes between bacteria, but similar processes are used to transfer genes into eukaryotic cells, like plants. And it turns out that some of this high-tech genetic manipulation is happening in some very unlikely places. This is the village of Chunamina in Zimbabwe, southeastern Africa. There has been no rain for many weeks. The village's crops of corn and millet have failed. The only staple left is cassava, its starchy, edible tubers safe underground, protected from the scorching sun. Victor Masona is a Zimbabwean plant biologist.
Cassava is the only standing crop. When we dug up a few tubers, I mean, it was like everybody was going for them, even before they were cooked, which means there is evidence of hunger. Everyone is hungry.
Cassava has brought Masona to this village. The plant is eaten every day by 500 million people in Africa and around the world. Westerners seldom encounter it, except in tapioca pudding. But here, it is the difference between survival and starvation. When something goes wrong with this crop, there's nothing left to fall back on. And something is wrong.
So this is the African cassava mosaic virus. You know, it's a disease that affects cassava, and you can see it by the crinkling of the leaves. You know, there's a mosaic on the leaves. All the shoots that come out have got the same disease. So you can actually spread the disease by propagating cuttings from this plant. So it can also be spread by whiteflies, sucking on this plant, and then they go and suck on to the next plant, and that way they transmit the virus from one plant to another.
Masona's collaborator is plant biologist Ian Robertson of the University of Zimbabwe.
Throughout Africa, this material is dug out of the ground. They see these symptoms, but they don't know it's the virus. So they'll break off pieces and they'll pass them to their relatives. And they'll plant them, but they'll plant them with the virus in the stems. So when the thing grows, it'll be full of virus, even fuller than this. And the difference between this plant and this plant is this one will give you at least 30 tons per hectare, and this one will only give you 6 or 5 or 4 or 2.
The villain is the tiniest of all microbes, a virus. Viruses are parasites. They enter animal and plant cells and use their victims' biological machinery to reproduce. Viruses are difficult to fight. Pesticides could help defeat this virus by eradicating its insect carrier, the white fly. But chemical treatments are too expensive and also might contaminate the water supply. Victor Masona uses another line of attack, one that will genetically alter the cassava plants.
Our work involves the genetic transformation of cassava for virus resistance. What this means is that we are trying to introduce genes into cassava. We hope that at the end of the day we will have genetically transformed cassava which is resistant to African cassava mosaic virus.
If you get the gene in, then we can produce for them Victor's kind of stuff, which is going to be virus resistant. Then we begin to solve the problem.
Masona obtained a grant to pursue his experiments at the International Laboratory for Tropical Agriculture Biotechnology in La Jolla, California. He fights one microbe, the cassava mosaic virus, with another microbe called an agrobacterium. Agrobacteria routinely pass their genes into plant cells. In a test tube, Masona mixes a solution of agrobacteria and genes from the virus. An electric current punches holes in the agrobacteria. The mosaic virus genes enter. The agrobacteria then transfer the viral genes to cassava plant cells.
You're trying to vaccinate the plant, right? So we are putting a gene from the virus into the plant, so it becomes part of the plant.
Masona gives the plant just enough of the virus so it can defend itself when the real virus attacks.
And the agrobacterium transfers the gene to the cassava cells, and then we take the cells, culture them, and grow them into plants.
After 5 months of careful nurturing, Victor has a finished product, a generation of genetically altered, mosaic virus-resistant cassava plants. The next step is to field test the transgenic plants in Zimbabwe's climate, with Zimbabwe's farmers, among Zimbabwe's whitefly population. Ian Robertson dreams of the day when Masona's plants will thrive throughout Africa.
I'll go through all this procedure of multiplying in the field, and in 3 years' time we'll have 200,000 for the next batch of people who need it. Then they'll get 30 tons per hectare every year, and their cousins will get it and their friends will get it, so suddenly we'll have 30 tons a year more or less permanently.
But it's not quite that simple. Genetic engineering is a powerful new technology. Its long-term effects are hard to predict. Zimbabwe's leaders are cautious. They're drafting regulations for the testing of transgenic plants. Masona cannot bring his transformed cassava into the country until the regulations are in place.
It can be very frustrating that the plants are sitting in my lab in California. I cannot proceed with the testing and the introduction of genetically engineered cassava if the regulations that guide these sort of introductions are not in place. So the people are hungry, but there's something that could help the situation, and yet I'm not able to bring it here because I'm not allowed to do so.
Biotech holds out the promise of enormous power. We can manipulate crops, animals, and even people. That's a dangerous power. So we need decent people to make the choices.
Scientists like Masona are sometimes accused of toying dangerously with Mother Nature, of trying to play God.
I don't think I'm playing God. I think if God would even be happy about that situation, because our aims are to help alleviate the problem of disease in cassava in this case, by obtaining crops that can be used by very desperate people. And I don't think that is bad.
Masona hopes he will soon be permitted to test his cassava plants in Zimbabwe. if they prove resistant to the cassava mosaic virus, a young biologist's manipulation of the power of microbes could nourish hundreds of millions of people.
To you is that plants or crops can also suffer from diseases. My research involves trying to put things into the crop so that, or into the plant, so that it doesn't suffer from the disease.
Perhaps Masona's attempts can only be successful if 21st century science is brought to everyone through education. When genetic transfer processes like transformation, the one he uses in his work, are better understood by all of us. Experiments with DNA transformation are also conducted regularly by Dr. So and her staff at Oregon Health Sciences University.
We do DNA transformation quite a lot in the lab, and we use the technique to genetically manipulate our bacteria, create mutations that we can follow for our studies, put in new genes that we can test for their virulence characteristics. So I can give you a demonstration.
Perfect.
I streaked out a strain of bacteria onto a plate, and these have been grown overnight. I'm going to swab these cells into these tubes and add DNA to each of these tubes and incubate bacteria with DNA and let the bacteria bind and take up the DNA. Now, the DNA contains a drug resistance gene, which has been inserted into a gene that we think might make our bacteria virulent. So we're actually mutating the virulence gene by inserting the antibiotic resistance gene right in the middle of it. The bacteria take up this DNA and insert it into their genome. They're going to be resistant to this antibiotic. These plates contain antibiotics, so bacteria that have taken up DNA will be able to grow on these drug plates. So after an hour of incubation, I've taken the bacteria and I've spread them on antibiotic medium, and then I've incubated the plates, and that's an overnight incubation. And here are some plates from yesterday's transformation experiment.
Okay.
First of all, we have the no DNA control. The bacteria were not incubated with any DNA, and so you see there's no transformation. They can't grow on drug plates. Here we have bacteria incubated with a large amount of DNA, and you can see that the plate is covered with bacterial colonies.
So they inherited a great deal of antibiotic resistance.
Well, that's right. The higher the amount of DNA, the more transformation events that take place. So a larger population of bacteria have now acquired the genetic trait. Here's a plate containing the smallest amount of DNA, and you can see that there are bacterial colonies on the plate, but there are fewer of them. We are using genetic engineering to try to understand how bacteria cause disease. In addition to that, scientists are now perfecting processes of DNA transfer to higher organisms based on these age-old bacterial genetics techniques. So in the case of farm animals, it is hoped that gene therapy can improve animal husbandry. And in humans, it is hoped that gene therapy can be the answer to a wide variety of medical problems.
For instance, we used to get the insulin used by diabetics from pigs like old Prince here. Now, pig insulin is close to human insulin, but not exact. Now, we can make human insulin by using microbes. By understanding the mechanics of microbial genetics, we can manipulate those mechanics for our own benefit, from improving health to turning the tide on world hunger. Over the 3.8 billion years they've been evolving and coping with changing environments, microbes have developed a variety of mechanisms for passing on genetic information and for survival. In the next episode, we'll see that it's a skill microbes have been honing since the dawn of life.
Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
For information about this and other Annenberg Media programs, call 1-800-LEARNER and visit us at www.learner.org.